All right, and some other news this week on the Inside the Ice House podcast. Host Lance Flynn wrapped up the biotech series with the final 2 episodes. And first, he sat down with Abbas Kazemi, CEO of Nimbus Therapeutics, a company that is pioneering the development of small molecule medicines that aim to transform patient care. Kazemi shared what is driving Nimbus to reshape medical practice and improve lives. Let's take a look.
You have to have the conviction on a target. You have to fundamentally believe we're going to change the course of medical practice for a patient segment, for a therapeutic area. But you also have to be careful of not chasing false positives. And what is a false positive? False positive could be we have an early signal in one patient, kind of like a biotech curse. And now you're going to spend money and time and keep going because we have changed an individual's life. The question becomes, was that a one-off case? How fast can you go? How much longer should you go? And that is where I think the false positives happen. You have a lot of companies that can really speak about chasing that false positive, and capital gets burned. And that's why I think right now investors are healthy skeptics of how much conviction do you have, but do you have the right discipline?
Glenn also welcomed Viswa Kaluru, founder and CEO of Inveda. The company is on a mission to unlock the untapped potential of nature's chemical code to accelerate the discovery of better medicines. And here's what he had to say about the ways that Inveda is helping chemistry experience its one next generation sequencing moment. Let's take a listen.
If historically this has been highly successful, and the idea is also deeply intuitive, right? Because living systems create molecules that are useful for living systems, then why did the industry move away from it? Why has the collective scientific enterprise only given us 300,000 compounds? And the short answer is that we didn't invent technology that allows us to take any sample and answer 2 really simple questions. What are the molecules in the sample, and what do they do? Right? In other words, chemistry never had its next generation sequencing moment. Today, you can take any natural sample, you can amplify the genetic material, and you can ask, what is the sequence of nucleotides? In other words, what are the genes? And then by comparing them to other sequences, ask what they might do. But there really isn't an equivalent for being able to crack the chemical code of a sample.
Our thanks again, of course, to Nimbus Therapeutics CEO Abbas Kazemi and Inveda founder and CEO Viswa Kaluru for those clips.